Dear students, welcome back to E-Pathashala. I am Dr. Sunil Kumar Onteru, a senior scientist in National Dairy Research Institute. Today we are going to discuss about the another lecture that is on HPLC in the paper Biochemistry. HPLC means high performance liquid chromatography. In this lecture, we are going to study about the principle of HPLC, the instrumentations of HPLC and the limitations of HPLC, and finally we are going to discuss about the supplements to the HPLC. Let us start our lecture with an instrument of HPLC, how it looks like. So, in this slide we are showing the typical HPLC instrument. You can see a computer screen and different pumps and column and also detectors. They are all the different parts of the HPLC instrument. To understand the basic concept and principle of HPLC, to understand the components and techniques of the HPLC, and to know alternatives to HPLC. In the good chromatography needs high resolution, a large number of periodical plates in a column, a low plate height and high values for the separation factor. So, how can we achieve such a good chromatography? In the coming slides, next slide we are going to discuss about this. So, one of the possibility for improving the performance of the chromatography is decreasing the particle size of the stationary phase. When we decrease the particle size of the stationary phase, that increases the surface area, that leads to an increase in the number of equilibrations or theoretical plates in a column. So, increasing the number of equilibrations leads to effective separation of the molecules with great resolution and other chromatography performance parameters. So, finally what happens? By decreasing the particle size, the performance of chromatography will be increased. So, this is one possibility to improve the performance of the chromatography. But there are some problems because of the small particle size. This we can see in the next slide. So, what are the shortcomings of the smaller particle size? So, by decreasing the particle size, there is an increase in the resistance to the mobile phase. So, because of the increase in the resistance to the mobile phase, there is a back pressure in the column. So, once there is a back pressure in the column, that results into the damage to the stationary phase, and finally there is a decrease in the eluent flow and the resolutions. So, these are the shortcomings of the small particle size that are used in the chromatography. So, what are the solutions for these shortcomings? In the next slide, we will see the solution for these shortcomings. The main solution to such problems due to small particle size is to select or prepare small particles that can withstand the pressures of at least 40 megapascals or high pressures. And also another solution is the requirement of high pressure to increase the flow rate. So, basically we need a small particles that can withstand the pressure, and another requirement is we need such an instrument which can produce high pressure to increase the flow rate. So, as by decreasing the small particle size, we are increasing the performance of chromatography, that is why it is called as high performance liquid chromatography, and also we are using high pressure to increase the flow rate, that is why it is called as high pressure liquid chromatography. So, HPLC principle is separation of biomolecules with high performance parameters under high pressure by using appropriate instrumentation. So, this principle we got based upon the previous analogy what we explained just now, that decreasing the particle size increases the performance, and to avoid any problems to the column we are increasing the pressure by using high pressure pumps. So, that is why the principle of HPLC is again the separation of biomolecules with high performance parameters under high pressure by using appropriate instrumentation. So, what we can see here, appropriate instrumentation. That means HPLC needs a special instruments with special characteristics. Let us see what are the HPLC instrumentations or the components in the next slide. So, in this slide we can see the HPLC instrumentation or components. You see in this picture we have the mobile phase reservoirs and we have the pumps, and also we have the gradient mixture to mix two kinds of mobile phases, and also we have sample injection valve which can be used to inject a sample into the column, and we can see a small column here that is called as guard column. Guard column is the column which is used prior to the main column just to avoid any impurities to enter into the column or with the main column. So, the guard column protect the main column by stopping some of the impurities not to going into the main column. So, then we have main column and also we have the detector, and finally we have the fraction collector. So, let us discuss all these components of the HPLC instrument one by one. Let us start with the mobile phase. HPLC mobile phase usually comprises of two or more solvents such as like water and acetonitrile for the separation of the biomolecules. These solvents should be highly purified and chemically unreactive. Why? Because the high purity is required if there are any impurities in that will interfere in the detection system. So, if these solvents are not pure, the impurities may interfere in detection system, especially at a wavelength of less than 200 nanometers. Basically, we need to use HPLC grade chemicals while doing HPLC. That means HPLC requires highly purified HPLC grade chemicals as mobile phase. For HPLC mobile phase, we need to do some of the activities. So, one of the important activity is degassing of the solvents. The solvent should be degassed before using them in HPLC. Why? It is important for those eluents with aqueous ethanol and methanol. So, especially whenever we are using aqueous ethanol and methanol, such kind of mobile phases, they should be degassed. Why? The important reason for this is we are using high pressure in HPLC. So, because of high pressure, that high pressure results in bubbling of gas or air present in the solvents, and those bubbles damage the stationary phase. That is why we need to remove the air bubbles from the mobile phase. So, that happens by degassing. So, there are several degassing methods for this purpose. These degassing methods are like warming, vigorous stirring with magnetic stirrer, vacuuming, ultrasonification, or budding helium gas through eluent reservoir. However, the most common degassing method is vacuuming. This is very common thing in case of any HPLC labs. We have the vacuum pump. By using vacuum pump and special instrument, we can degas all the solutions by using 0.4 micrometer filter or anything 0.2 micrometer filter. The selection of the mobile phase mainly depends upon the type of separation and the components in the sample. Usually, polar and non-polar solvents are used in certain ratio to obtain effective separation of sample components. The mobile phase solvents should not interfere in the detection system. So, by taking all these points in mind, we have to select the mobile phase. So, once we have selected the mobile phase, there are different techniques for HPLC. So, what are those different techniques for the HPLC? So, basically the techniques based upon the mobile phase are two kinds of techniques. The first technique is the isocratic elution. Isocratic elution means mobile phase composition is constant throughout the HPLC separation. That means the composition will not change. It will be, we will use one particular solution, and that particular solution will be using continuously throughout the HPLC experiment. So, what are the advantages for such kind of isocratic elution? The main advantages of this isocratic elution is they are used for the simple mixtures, and another advantage is by using the same composition, the system and column are equilibrated all the time with the same composition, so that the stationary phase will not suffer from any fast chemical changes by changing the mobile phase. So, by using the same composition, the stationary phase is always exposed to same kind of chemical composition. So, your separation will be very nice in this isocratic elution. But however, we have to use gradient elution in some of the experimental conditions. Gradient elution means mobile phase composition is gradually changing in the ratio of polar to non-polar compounds during the sample run. So, what is the advantage of this gradient elution is this can be used mainly for the complex mixtures, but for the simple mixtures we can use isocratic elution, for the complex mixtures we can use the gradient elution. In addition to the different techniques of HPLC by using mobile phase, the technique of HPLC can be classified into another two categories, that is the normal phase HPLC and reverse phase HPLC, based on another criteria that we will see in the next slide. So, in case of a normal phase HPLC, that means the stationary phase is more polar than the mobile phase. So, in the previous slide we have seen that the mobile phase composition is the same, then it is called as isocratic elution, and if mobile phase is different at different timings, that is called as gradient elution. But in this slide we are seeing that the stationary phase, if it is more polar than the mobile phase, then it is called as normal phase HPLC. The important more polar stationary phases are alumina or silica. In this kind of HPLC technique, the first compound that is coming out of the column is less polar. Why? Because the stationary phase is more polar. So, more polar compounds will be attached to the stationary phase. That is why the more non-polar compound will come out of the column in case of normal phase HPLC. That is why the first compound to be eluted in normal phase HPLC is less polar or more non-polar. The another technique of HPLC is reverse phase HPLC. Reverse phase HPLC means the stationary phase is less polar than the mobile phase. When it is less polar than the mobile phase, the non-polar compounds are attached to the reverse phase. So, non-polar compounds are attached to the stationary phase. That is why the more polar compounds will be eluted first in case of reverse phase HPLC. So, what are the stationary phases that have less polarity than the mobile phase are? Examples are alkyl or aliphatic or phenyl bonded phases. They have less polarity than the mobile phase. So, in the reverse phase HPLC, the first compound to be eluted is more polar, and the retention time for a compound increases with decreasing polarity of a particular species. That means the more polar compound will come first, its retention time is less, and also the less polar compound will come last. That means its retention time in the column is for long time. So, that is about different techniques of the HPLC based upon the mobile phase and also based upon the stationary phase. So, let us see what are the properties of the stationary phase in case of HPLC in the next slide. HPLC stationary phase particles they are mainly rigid rather than soft gel. as in open column chromatography. Normally stationary phase particles are the soft gels in case of open column chromatography, but in case of HPLC, these particles are solid rigid particles. Why? Because they have to sustain high pressure in the column chromatography. I mean HPLC, that is why the solid particles are more rigid in nature. And second important property is these solid particles, they should have spherical in shape. That means there should be spherical particles with uniform size to reduce space for diffusion. So, spherical particles, when it is small and also spherical, they reduce the space for diffusion. So, what are the types of stationary phase particles? There are mainly 3 types of stationary phase particles. The first type is called as microporous particles, and second type is called as pellicular particles, and third type is called as bonded phase particles. Let us discuss about these particles one by one. You can see in this slide, the microporous particle looks like a very round spherical particle containing so many pores on its surface. So, this is a microporous particles. It is the diameter is 5 to 10 micrometer in diameter of this particular particle, and these particles are mainly composed of silica or aluminum, and they contain microscopic pores. This is very important in case of microporous particles. So, these microporous particles contain microscopic pores which run through the particle. Because of that, the surface area for the interaction between the mobile phase, stationary phase, and also the sample components will be high. That is why these particular particles, the separation will be efficient. The next stationary phase particle is pellicular particles. Pellicular particles are also called as superficially porous particles, and these superficially porous particles, they have a diameter of 40 micrometer in nature, and these particles have an inert core on which the pores are coated. The inert core is made up of glass or some other hard material, but the porous material could be made up of polymethacrylates or polystyrene divinyl benzene. So, these are the materials by which the porous material is made up of, which is on the rigid hard inert material. These particles display excellent pH stability with high mechanical stability. That is why these kind of stationary phases containing pellicular particles can be used at the wide range of the pH experimentations. As we have the inert core inside, the mechanical stability for this kind of particles is also very high. In the next slide, we will discuss about our third stationary phase particle, that is called as bonded phase particle. These particles also contain an inert core made up of the silica, and the stationary phase is chemically bonded on that inert core by different linkages, for example ester bonds. And what is the example for these bonded phase particles? One of the best examples for the bonded phase particles, which is commonly used in the reverse phase chromatography, is C18 column. So, silica C18 column is commonly used in reverse phase chromatography for the separation of peptides or proteins. So, let us see the formats of the HPLC columns in the coming slides. In the next slide, we will start with the conventional HPLC columns. So, basically the HPLC columns are those structures that can hold a stationary phase. There are majorly 3 kinds of columns, that are the conventional HPLC columns, capillary columns, and nanobore columns. Let us discuss one after another. The first one is the conventional HPLC columns. Conventional HPLC columns are made of stainless steel Why we have to use stainless steel? Because to withstand high pressures like 50 megapascal pressure, and these kind of columns are generally 3 to 25 centimeters in length. The internal diameter in these columns is nearly 2 to 5 millimeter in diameter. The flow rate of mobile phase in these columns is 1 to 10 ml per minute. Let us discuss about the next column format in the next slide. The capillary columns are thin columns. The inner diameter for these columns is 100 micrometer to 1 millimeter, and the flow rate in these columns is very slow, like 0.4 to 200 microliter per minute. And these are very important capillary columns used especially in case of mass spec and HPLC combination kind of instruments, and further thin columns are present, and those are called as nanobore columns. So, the nanobore columns are further thinner than the capillary columns, and the inner diameter of these nanobore columns is 25 to 100 micrometer in nature, and the flow rate in this particular nanobore columns is 25 to 4000 nanoliters per minute. The nanobore columns are mainly helpful to use at the interface with the mass spectrometry. These columns are important in situations to analyze minute quantities of the proteins, where we use minute quantities of proteins, especially whenever we are using the proteomic studies, we will use the minute quantities of proteins. So, these nanobore columns are mainly useful in case of the proteomics studies. So far we have discussed about different stationary phases, and also that different stationary phase column formats, and also the mobile phases and the different techniques of the HPLC based upon the mobile phase and the stationary phase. Let us see how the stationary phase is packed in the columns. Now, just now we have discussed about the stationary phase, and also we have discussed about the stationary phase column formats. So, we need to fill the column formats with the stationary phase, that we will discuss in this coming slide, that is the stationary phase packing. The stationary phase is packed into a HPLC column by a specific procedure called as high pressure slurring procedure. In this procedure, the stationary phase and solvent for slurry need to be in equal density. So, the stationary phase and the solvent which are used for the slurry, they should be in equal density, and the slurry can be pumped into a column by high pressure. That means, for packing the HPLC column also we need high pressure, and the density in which the stationary phase is made up of, in the solvent in which the stationary phase is made up of, that slurry also should be with equal density with the stationary phase density. So, by high pressure and high pressure slurring procedure, a dense continuous bed of stationary phase without any cracks and other imperfections will be formed. So, our main motto of the packing of the stationary phase column is there should not be any imperfections in the stationary phase column. Entire column should be uniform with the stationary phase particles. That is why we need to use a specific procedure, that is called as high pressure slurring procedure. So, that is all about the columns and also the stationary phase and also the mobile phase. And let us shift to other components of the HPLC experiment or HPLC instrumentation. An important requirement in HPLC is the flow of mobile phase, and that flow of mobile phase should be very stable, should be pulse free. There are two types of the pumps. These pumps are the constant pressure pumps or the constant volume pumps, or we can also call it as a constant displacement pumps. What are these constant pressure pumps and what are these constant volume pumps? We will discuss in the next slide. Constant pressure pumps maintains constant pressure irrespective of the stationary phase resistance to mobile phase flow. If the resistance increases in stationary phase, the flow rate will be decreased automatically to maintain the constant pressure. So, the main motto of this pump is to maintain the constant pressure. Accordingly, the flow rate may be changed according to the pressure situation in the stationary phase. The constant volume pumps maintain constant flow rate through stationary phase, and the pumps increase the pressure if there is an increase in stationary phase resistance. If there is a resistance in stationary phase, this pumps also increases the pressure in the column. So, however, increasing pressure is within the preset limits of the pump. So, this pump have certain limits, and that increasing pressure that is within the preset limits of the pump. If the pressure is required more than the preset limits, then what happens? The pumps are automatically inactivated by safety cut mechanism. So, moreover, these constant pumps, in the next slide we can see what are the properties of constant pumps. So, these constant pumps are more popular in HPLC. So, when we compare between the constant pressure pumps and constant volume pumps, the constant volume pumps are more popular in case of HPLC. So, one of the examples of constant volume pump is a reciprocating pump. So, this is shown in this particular picture how the reciprocating pump looks like. In this reciprocating pump there is a piston, and this piston is useful to deliver a fixed volume of solvent onto the column in repeated cycles of filling and emptying. That means it releases the constant volume in pulses. Generally, there should not be a gap between the pulses of the mobile phase. Therefore, in order to avoid pulses while the flow of mobile phase, the pulse dampeners are built into the pumps. So, by using this pulse dampeners, the pulses effect can be nullified. So, there will be constant flow of the mobile phase. So, this is about the pumps in the HPLC instrumentation. So, the next slide we will discuss about how to apply the sample and what is the injector in case of HPLC. The most common injector in the HPLC is the loop injector. That means it has one loop which is operated manually. So, if you see the pictures here, there are two pictures. One is on the left hand side, that is A, another is the right hand side, that is a B. The left hand side picture represents the loop injector in the loading position, and the right hand side it is shown in the injecting position. Let us observe the connections of tubings in the loading positions. So, in this case you can see there are numbers of 1, 2, 3, 4, 5, 6. There are 6 kinds of ports, and where there is a connection between the first port and the second port, and in this case the eluent is coming from the pump to the first port, and through tubing it is connected to the second port, and through which it is going to the column. That means the mobile phase is passing from the pump to the column directly, and there was a connection between the port 3 and port 6, and there was a connection between port 6 and port 5, and there is no connection with the port 4. So, there was a connection between port 3 and port 6, that is the loop, and this loop has a fixed volume, and in this port only we need to inject a sample. Once we inject the sample, for example, if this loop has 20 microliter volume, once we inject say for instance the 40 microliter of the sample, then this loop can hold only 20 microliter, and excess of the 20 microliter it will pass on from the port 6 to the port 5 and port 5 to the waste. So, this loop contains only 20 microliter in that case if its volume is 20 microliter. So, in the injector position we have to turn the knob which is at the center to the right hand side. By turning this knob, what is happening you can see here. Here there was a connection that is established between the port 1 and port 6 and another connection is established between port 2 and port 3. So, by doing this what happens is the mobile phase is coming from port 1, it enters into port 6 and it passes on through the loop continuously and reaches to the port 3 and through port 3 through the established connection to the port 2, then the mobile phase pass on to the column. So, the mobile phase now it is passing through the loop to the column. That means it is taking entire sample whatever we injected at the loop 3 and that entire sample is transported to the column. So, that is a principle in case of the loop injector that is used in HPLC. So, the next slide we will discuss about another component of HPLC, that is guard column. Guard column is a small column between the injector and main HPLC column, and this column is mainly used to protect the main column from impurities. So, whatever the impurities present in the sample, those impurities are captured into the guard column so that the pure sample will go and enter into the main column. So, that is the main advantages of the HPLC guard column. So far we have discussed about the pumps, we have discussed about the mobile phase, we have discussed about the stationary phase, we have discussed about the stationary phase column formats, guard column and injector. Let us discuss about the HPLC detector which is coming after the column in the HPLC instrumentations. So, there are several kinds of the detectors in this slide. If you can observe that there are almost 9 kinds of HPLC detector. So, these 9 kinds of HPLC detector are majorly classified into 2 categories based upon the detection mechanism. The first category is called as optical detection mechanism and second category is called as electrical detection mechanism. So, what are those particular detectors in each category? In the optical category, the detectors are variable wavelength detectors, scanning wavelength detectors, fluorescence detectors, refractive index detectors, evaporative light scattering detectors. But in the case of electrical detection, there are 3 kinds of category, that is the electrochemical detector, conductivity detector and mass spectrophotometer detector, and finally the NMR spectrophotometer detectors can also be used. These last 2 detectors, these are the mass spectrophotometer detector and NMR spectrophotometer detector. Generally these detectors are present in those instruments where there is a combination of HPLC and mass spectrophotometry and combination of HPLC and NMR spectrophotometry. In the next slide, we will discuss about how to choose which kind of HPLC detector system we have to use. So, in this slide, I am showing one algorithm by which we can detect different HPLC detectors. So, based on the compounds and their properties, we can choose a specific detector among different detectors. This particular algorithm helps to select the best detector for HPLC analysis. For general analysis like quality control analysis of medicines or food with sufficient quantity of the samples, if the test compounds have the property of UV absorption, then we can use UV or UV visible or DAD detectors in this particular case. If these compounds do not have the property of UV absorption, in such samples we can use RI detector or EL detector, SD detector and CAD detectors. Suppose we need to analyze rare biogenic substances like environmental contaminants, agrochemical residues, etc. in that, and also the concentration of which is very low. So, in that case we have two options. One is either UV or UV visible or fluorescent detectors if there is a possibility of derivatization for those compounds. If those compounds have the property of electrical activity, then we can choose ECD detectors. Suppose we need to analyze the inorganic ions in our samples, then we have to use the CD detectors. So, based upon this algorithm, we can choose what kind of detector we have to use in HPLC based upon the type of compounds which we are going to separate. Dear students, so far we have discussed about the principle of HPLC and instrumentations of HPLC and techniques of HPLC, and in the instrumentation we have discussed about the mobile phase, stationary phase columns and pumps, injector and God column, and finally we have discussed about different detectors and the algorithm that can be used to choose a best detector in HPLC experiment or HPLC instrumentations. But there are certain limitations for HPLC, and these limitations and what are the alternatives for HPLC limitations that we are going to discuss now. The main limitations of HPLC are there is a high cost of generation of high pressure, and high pressure imposes some constraints on instrumentation. HPLC also has relatively low flow rates, so the experiment is very time taking. So, these are the main three limitations for HPLC, and these three limitations can be overcome by decreasing the pressure and increasing the flow rate for comparable resolution of analytes by HPLC. So, this can be done by another kind of alternative for HPLC, that is called as perfusion chromatography. So, another solution for these limitations are increasing the instrumentation that can withstand a high pressure by considering cost economics for better resolution than HPLC, and such kind of format is called as, one of the alternative for this is UPLC, and this is another alternative format of HPLC. Another limitation of HPLC is especially useful in the separation and analysis of low molecular weight molecules, but it does not usually useful for purification of complex biopolymers. For example, proteins. If you want to purify the proteins by HPLC, it is not that much good in the, especially the proteins in the active form. So, for complex biopolymer purification, HPLC is not the best method. What are the reasons for it? The first reason is some HPLC formats like reverse phase HPLC needs organic solvents. So, these organic solvents, they denature the proteins. That is why these proteins, we may not get the active form of the proteins by this particular chromatography. So, another reason is, although large capacity HPLC columns are available, they are very expensive. So, small capacity columns are quite common. So, what is the probable solution for this kind of limitation for the purification of the biopolymers? One of the another alternative for the purification of biopolymers is FPLC, that is a fast protein liquid chromatography. So, now we are going to discuss about these HPLC supplements in the coming slides. So, as we just now discussed while discussing about the limitations, so these are the three HPLC supplements, that is the perfusion chromatography, ultra performance liquid chromatography, that is UPLC, and fast protein liquid chromatography, that is FPLC. Let us talk about these chromatographies one by one. So, perfusion chromatography means with small particle size, high resolutions can be possible in short times with high flow rates. This happens in case of perfusion chromatography. So, in this chromatography, the stationary phase particles contain not only longitudinally pores throughout the particle, but also diffusive pores that are connecting the longitudinal pores. This can be seen in this picture in this particular slide. You see the conventional chromatography picture, it is like longitudinal pores are present, but in case of perfusion chromatography and these longitudinal pores, they are also connected by the diffusive pores in a horizontal way. The best example for this kind of particles are the porous particles, which are made up of polystyrene divinyl benzene, and these particles, they have size is usually 10 to 50 micrometer, and in these particles the surface area is large due to pores, so in which the stationary phase is located. This kind of chromatography can be performed by normal HPLC instruments. How can we obtain the high resolutions with high flow rate? So, before going to those details, let us see what kind of pores are present in the porous particles in the next slide. So, there are two types of pores present in the porous particles. One is called as large through pores for rapid flow, and another one is diffusive pores for connecting the large pores. This is very important because mathematically we can prove that there is no effect of flow rate on the theoretical plates in the coming slides. So, these kind of pores, they provide large surface area and the stationary phase is coated on that surface area, and the same type of HPLC instruments can be used with less back pressure that we discussed just now. So, how can we get the better resolutions or better chromatography by using high flow rates in the perfusion chromatography? For knowing that, first of all we need to recollect our knowledge on the theoretical plates heights. Okay, this we can see in the next slide. So, in this slide you can see the theoretical plate height is inversely proportional to the diffusion coefficient of the mobile phase. So, in this mathematical expression you can see that H equals to C prime and multiplied by the C prime is the coefficients of stationary phase particles and dP is the particle diameter, that is dP square here, multiplied by flow rate of the mobile phase and divided by diffusion coefficient of the mobile phase. So, basically what we need to understand here, the theoretical plate height is inversely proportional to the diffusion coefficient of mobile phase. So, let us see in the coming slides how it is mathematically related. First of all, we understand this in this slide, what is the diffusion coefficient. So, diffusion coefficient dm equals to capital D plus V pore means multiplied by dP by 2. Here V pore means flow rate within the stationary phase. dP means particle diameter. So, the first expression is capital D, that is called as diffusion component, and this diffusion component is dominated in case of, or diffusion component dominates in HPLC and also FPLC due to low flow rates. The connective component is the V pore multiplied by dP divided by 2. That means V pore means the flow rate of mobile phase within the stationary phase, and dP means it is the particle diameter. And in case of HPLC, this diffusion component dominates, and the same case in case of FPLC, where the diffusion component dominates, but in case of perfusion chromatography the connective component dominates. So, in the next slide further mathematically we can see this. At high values of the flow rate, the connective term is much more important than the diffusion term. So, we can nullify the diffusion term. Then the diffusion coefficient of the mobile phase is approximately similar to the connective term. So, connective term is V4 multiplied by dP by 2. But there is a proportional relationship between V4 and the V, that is the flow rate. So, V4 equals to a particular constant multiplied by the flow rate of the mobile phase. So, in the next slide we will see further mathematical expressions by substituting the dm with constant multiplied by flow rate multiplied by dP. So, if you see this particular value of dm in case of theoretical plate height, the V and V are cancelled. That means the theoretical plate in perfusion chromatography is not dependent upon the flow rates. So, whatever may be the flow rates, we can separate the molecules in the perfusion chromatography because the connective component is the most important thing in case of the perfusion chromatography. So, overall here by substituting the diffusion coefficient of the mobile phase with the connective component, the height of the theoretical plate become largely independent of the flow rate V, but it dependent upon the particle diameter, because when you see the particle diameter is in the numerator. So, similarly interparticle diffusion becomes unimportant relative to the intraparticle convention. So, therefore, perfusion chromatography is an alternative to HPLC to obtain results with the high flow rates without losing any resolution. So, coming to the next slide where we will discuss about another alternative of the HPLC, that is a UPLC. So, UPLC is called as ultra performance liquid chromatography. What happened was the commercial company, that is Waters Corporation, that company developed another kind of chromatography, that is the UPLC, and in which the particle size is decreased and then the capacity of the instruments is increased. Because of that, what happened? The particle size is decreased to 1.7 micrometer diameter. That means the stationary phase particle size is 1.7 micrometer diameter. The stationary phase particles are also made up of bridged ethyl siloxane silica hybrid. This particular material can withstand a high pressure, and the chromatography by using this kind of UPLC, it is 10 times faster than the conventional HPLC. What they have done basically, they decreased the particle size and increase the capacity of the instruments that can withstand the pressure. That is the simple thing. So, by doing that, the efficiency of HPLC is increased and faster, and that is called as UPLC. So, we can appreciate the separation of the molecules and the fastness of the UPLC in the next slide. So, in this slide we can see 4 kinds of pictures, and all 4 kinds of picture look like similar, but the difference in them is only at the retention time. So, what is the difference in that? We have used here a same compound, one we analyzed by HPLC, another one is analyzed by UPLC, or simply that we have analyzed this particular compound in that stationary phase where the stationary phase particle diameter is decreasing. If you see the top left hand side top figure, in which the stationary phase contains 5 micrometer size of the stationary phase particles, in that case the first peak is coming at the 4 minutes time. Then once the stationary phase particle size is decreased to 3.5 micrometer, the retention time of the first peak we can see at 1.5 minutes. But if you decrease further the stationary phase particle size to 2.5 micrometer, the retention time is further decreased to 0.8 minutes. Then if you further decrease to the stationary phase diameter size to 1.7 micrometer, the first peak we can see at the 0.5 minutes. That means the entire analysis in the first instance where 5 micrometer diameter stationary phase took 10 minutes, but the same sample took only 1 minute when the stationary phase particle size is decreased to 1.7 micrometer. So, this kind of chromatography is called as ultra performance liquid chromatography. In the next slide, we are going to discuss about another alternative for HPLC, that is called as FPLC, that is a fast protein liquid chromatography. Fast protein liquid chromatography, it is mainly useful for the biopolymer separation. For example, protein separation or DNA separation. And the major difference between the HPLC and FPLC is lower pressure in case of FPLC when compared to the HPLC. And in this FPLC also we can use reciprocating pumps, but reciprocating pumps should have 2 pistons, and 2 pistons can be used in these reciprocating pumps to avoid the pulses. The columns in this case of FPLC are made up of glass or plastic because of the low pressure, because we do not need a higher pressure. So, we do not need any steel columns and all in case of FPLC. We can use glass or plastic column, and stationary phases are usually compatible with the aqueous buffers and salts, because we are going to purify the biopolymers in active form, and usually biopolymers are in active state in case of aqueous buffers. Overall, in this particular lecture, we have discussed about basic concept and principle of HPLC, HPLC instrumentation or components, and HPLC limitations and supplements. Dear students, I hope you enjoyed this lecture, and I consider that you understood the basic principles of HPLC and instrumentations of HPLC and the techniques of HPLC, limitations of HPLC, and the alternatives for HPLC. If you want to read further, I suggest 2 important books. One is the Wilson and Walker, that is the title of this book is Principles and Techniques of Biochemistry and Molecular Biology, and another book is Physical Biochemistry, Principles and Applications by Sheehan. I think there are many other internet resources that you can check for this particular topic to understand much better, and I hope we will see next time in another lecture with another topic. Thank you very much.